I am Silvia Sanchez-Martinez. I am a senior scientist in Thomas Boothby's lab in the molecular biology department in the University of Wyoming. Tardigrades, they are microscopic animals. They are segmented. They have 8 legs. They have superpowers. They can survive in extreme environments. For example, they can survive high temperatures over the boiling of the water. They can survive freezing. They can survive high pressures, like living in the bottom of the ocean, or when we send them up to the space, the vacuum of the space. They can also survive like about 1,000 more times radiation than you and me can survive. They can live for weeks with little oxygen. Very important thing for our research is that they can survive nearly complete dehydration. They can survive with even only like 1% of water. So when tardigrades start losing water and they start dehydrating to that 2% of water, they start curling up into a ball-like structure. And in this ball-like structure, they become ametabolic. So their life processes, they cease. They can stay in this state for days, weeks, months, years, and in some cases, even decades. Once water comes into their environment and they start getting rehydrated, their metabolism returns. So in about half hour to an hour, you see them like running around the dish like nothing has happened, and they return to their normal life processes like eating, reproducing, like nothing has happened. What we do in the lab is like try to study these superpowers and then learn as much as we can from them, from the tardigrades, to be able to apply that for human health, for example, or for questions that humanity may need to answer, like the preservation of pharmaceuticals or biologics. These tardigrade proteins form gels. Forming a gel is a characteristic of proteins, so there are proteins like gelatin, collagen, they form gels, so it's not an unusual property. Usually, the forming of the gel is linked to a function. In our case, when we were looking into how this protein makes or forms the gel, what we find out is like as it happens in the tardigrade, the protein is able to slow down metabolism in human cells. So if you trap molecules inside of these gels, they slow down the processes of what you are encapsulating inside the gel. So we think that these proteins are involved in that process. So one applicability of these proteins that gel and slow down metabolism could potentially be in organ transplantation. So when you take an organ from a body, like certain processes carry on, and you have like an hour or so, like what is called the golden hour, to put this organ back into a new recipient. So we could apply potentially these proteins that form gels and slow down metabolism for these life-saving applications. The same thing in war zones or places with natural catastrophes where there is trauma. So if you could slow down the processes that start after trauma until you can get proper care. Now the tardigrade has more than one of these proteins, so we are studying one, but there are 11 more, so we are just learning little by little what one can do and going from there.